All right, so let me actually zoom out and show y'all what this new animal of the week looked like. Here is the new species. So this is a parasitic wasp. It's a male wasp that's shown. The thing about it is that the colony has males integrated to it. So this has no colonies, Chris. The life of these is mostly solitary, where they end up, just they land and they mate and then they infect and they move on. There's not really like a social system as far as we're aware with parasitic wasps. However, Chris, that could just be because the question hasn't been asked the right way, which is usually the case for these things. Sometimes the terminology, Chris, is called asocial, which means lacking any social anything. I don't like that description because it suggests that these animals can have no sociality, and that we know is simply not true. These animals have to have some kind of social interaction in order for them to be able to interact, to mate, to interpret signals from one another. That is just a standard piece of their biology for it. But it could be that it hasn't been asked in the right way. And easy to interpret, right? Drosophila, so fruit flies, are hosts to the first described parasitoid wasp of adult flies. Y'all, everything you need from the paper is in the title. Fruit flies are the hosts to a type of parasite that infects adults. And this is the first one. Like, nicely done title, right? So Shark, here's the catch though. There's plenty of parasitic wasps that parasitize fruit flies. That's nothing new. In fact, I worked with them during my undergrad. They parasitize the larvae of the fruit flies. Where this is a big catch is that these animals are parasitizing the adults. So let me show you. This is what we looked at for, honestly, y'all, these were discovered, I believe there's papers all the way to the 50s about fruit fly larvae being attacked. So in this video, that is a parasitic wasp located right here. And that parasitic wasp is going to be infecting a fruit fly larva. So the current, up until this paper was released, Shark, the literature even, if you were reading about what was the science behind response to parasitic wasp and the fruit flies, the very common thing that we would see in the literature, and I wrote this too in our publications, is that parasitic wasps are no threat to adult fruit flies. But so, again, in our papers, we would be writing, we don't know. There is no adult threat right now to fruit flies. So we were measuring behaviors that the flies would do as a function of being near these wasps. And it would always be any behavior they're doing has to be because they're trying to protect their babies because they aren't under threat. And this is one of those wasps here. So this is one, you can see these little wiggling things up in the top corners. Those are larvae. So those are fruit fly maggots that are crawling around and eating the food, and that's what the wasp is sensing. Is this a recent change in wasp behavior or just a recently discovered observed behavior? No. So Jay, this is the behavior of a species of wasp that parasitizes fruit fly larvae. The wasps that parasitize adult fruit flies that this paper is discussing is a brand new species. It is not a change in existing behavior, but rather a brand new animal that had never been described before. And the reason it's a big breakthrough is because people didn't think that there were animals that could be infecting adult fruit flies. And again, so it's not just a change and that's a good point, Jay. It's not a change in behavior of existing animals that now they're going to start infecting adult fruit flies. It is something totally brand new that no one's ever seen before. The shift from infecting larvae to adults would be a big change, not just on the molecular level, because parasitic wasps have venoms, and these venoms are specific to their hosts. And so in this case, it would be to fruit fly larvae that they're trying to take out. So this is the old one. You can see how it's going around these larvae, and you can kind of make out they've got the ovipositor label. So that's what Corn Relish was saying earlier. That is a needle-like thread that it holds the venom and an egg. And so that piercer injects into the host, whether it be a larva or now an adult, injects an egg and venom. Attacking adults would be a lot more difficult than larvae. It would be different. So the reason it's different is because, Jay, you have to infect something that's actively moving around in a different way than the larvae. So, Jay, in order to infect an adult, the adult infection is either you're having to bore through the exoskeleton, or you lay an embryo on the outside of the animal, and then when the larva hatches, it eats its way into the host. But right, that's a very different approach than what you're seeing here, which is the infection of a fruit fly larva. So that's what we're seeing here. You can see the ovipositor is stuck into the host, the fruit fly. So she's injecting right now venom and an embryo. So corn, they can feel this, as you can see. So this behavior that this larva was doing, the wiggling away, is actually, so normally in nature, you have the Z plane. You have the Z axis, which you can move in, meaning you can go up and down, right? In this dish, these larvae can't go up and down, so you see that there is scooting in one direction. The other thing that these larvae will do is actually roll around. There is a rolling behavior that they do. So, Jay, there are physical defenses that they can do, like rolling. They also have an immune system. So this is not 100% successful. This does not mean that the wasp is going to be hatching out from the inside of this fruit fly. There's a chance of it, but there's a lot of different factors at play. So this species of wasp is called Leptoplina ballardi. Ballardi is a specialist on Drosophila melanogaster. There's also generalists like Leptoplina heterotoma. That one means it can infect a variety of fruit fly species and be successful. So if you take the specialist and you force it to infect something that it doesn't specialize on, the fruit fly is more than likely going to win because its immune system, it's just like the animal's not primed to fight it. And so it's usually means that the venom that wasp is using is specialized to that host. Now, heterotoma, the generalist, uses a more, as the name suggests, general venom. And what that means is it's a way for the venom to overcome a lot of different hurdles. Heterotoma venom, what you can do is if you pop open the venom gland, there are molecules in the venom that pop open cells. So you can imagine the immune cells of these animals, they actually have to end up catching the individual host, right? Like here's the parasite as an egg, and then the immune cells surround it and kill the egg. But if you pop open all the immune cells, there's no immune cells to kill the egg. And then if the wasp egg hatches inside the larva and you have a wasp larva swimming around, the immune system's out of luck and the wasp is going to win. What is the conversion rate? How well does it work? It's very host specific, Jay. So the new species, they did give us those measurements. So we'll talk about what the metrics are for the new species, but it's honestly, Jay, anywhere from if you take a non-specialist and you just give them a wide range of hosts. I did that experiment with heterotoma, so that's the generalist, and I gave it 30 different species of fruit fly to infect. And there was everything from 0% to 100% infection rate. On the specialist, usually that the one that's specializing on a host is like between 60 and 70% successful. So it's not going to be all the time that they win, but a lot of the times that will be the winner. And so it just, again, it depends on what particular host parasite interaction there is. How does the venom work, and availability too. There's an interesting catch here as well, Jay, where if you look at this wasp, you see that ovipositor sticking out. That ovipositor can actually tell if the host is already infected. There's sensors on that ovipositor to when she sticks it into a host. The sensors will tell her, hey, there's already someone in here, or there's not. But what makes it maybe a little bit less crazy, Corn, is that it's species specific. So she can only tell if there is something in there from her own species. She can't tell if there's a different species member in there as well. So it's not just that someone's in there, it's someone's in there, but they have to be from my species for me to be able to detect. That's the catch here. If you dipped a teeny tiny dart in the heterotoma venom, use the tiny blowing thing or use a bathroom. So where it wouldn't work, Sam, is that would not work on the adult. Adult cells they're just not primed, because what it does, the venom for heterotoma, is it binds to a type of cell that's specifically in the larval host and not the adult host. So the venom just couldn't bind to the right thing in the adult. But I like where your head's at. Sam. Sparky, what if the fly dies before the larva is grown? Then the wasp also dies. So yeah, Jay, when you show adult fruit flies a wasp, their offspring that they lay are now immune primed. So their immune system is going to be better than the ones whose parents did not see the parasitic threat. So that is along the lines of your question that's been shown to be a feature. It is not confirmed, right? There are many different avenues that would need to be checked and adjusted for, but I think it's really cool there's something like that. So there is an epigenetic effect. It's just, with your question, it's if you survive, are you any better for that next generation? That's the one that hasn't yet been asked quite in the right way. So it's almost like the precursor, right? Like, is there a way for the moms maternally to prime their offspring for have a better immune response? That's a yes. So the second part would be, that other one. They do, Sparky. So the fruit flies see the parasitic wasp, and they react to it as a threat. Now, that we don't know yet for the new species, right? This species of wasp, Sparky, only infects those embryos that are like at the larval stage, right? These are no threat to the adults. The new species infects adults only, so you might predict that there might be differences in behaviors. This paper that we're going to dissect is just the initial study of, hey, there's this brand new species, this is what happens, and we'll get into it, but it's like a very initial experience and description that's going in. Hopefully that makes sense, Sparky. Is the stress response from the parent fly that primes the immune response in the next generation. It's some kind of, so Jay, it goes from the visual system, so the adults see the threat, they then signal the ovaries. So there is a neuropeptide that changes in the brains of these animals. It gets decreased in their brain. It's called neuropeptide F. It goes down, and then the ovaries are fundamentally changed, like the embryos hanging out in the ovary. We don't know if it's just general stress. There might be general stress there too, but we did figure out what neuropeptide was involved. And then what ends up actually happening is there's a gene that's turned up high in these embryos. And because they're turned up high in the embryos, it's like now they're primed and ready to go. And actually, Jay, what's cool is you can artificially increase that one gene, that one thing that you're like, maybe it's this thing that I think is priming the animal's immune systems. You could artificially crank that up without ever seeing a wasp, and then you have the same result. Like that artificial crank up primes the immune response as well. So we were able to actually figure out what gene it was. Now, how that gene is modified in the genome to have that happen, that's still what we call that black box, that mystery box, right? You go from like the brain, there's a neuropeptide that's decreased, and you finish with an embryo that has an increase in this particular genetic element, right? Everything in between is a question mark. So that would be the next step to figure out, okay, what is the signaling cascade? Like, what is this affecting this, affecting this, affecting this that actually led to something like the outcome of a better immune response? Again, I thought it was really interesting that there's so many of these wasps all over the US. Based on the description that they were talking about, I did not think that they'd be all over the United States, right? It's the first example of a parasitic wasp infecting an adult fruit fly. They are saying it is everywhere, right? It is everywhere under the sun in the US. Why did it take so long to identify? It might have been one of those that you just haven't asked the question the right way. You haven't done any of the askings. You haven't done the right type of examination. It's still surprising, y'all, I think. In the paper, they actually have, again, I think it's super clearly written. One of the reasons why I like this. We document its life history, unique host interactions, including its egg laying into and larval emergence from adult flies, and provide protocols to raise the wasp from wild-caught host flies. And then they even say it's the emphasis for ongoing research investment in insect biodiversity and systematics. A parasitoid research continues unusual biology supports fundamental mechanisms. And Jay, this is what you said, insights to immunity, metabolism, ecology, evolution, and behavior. And 100%, Sam, that's also a kickoff from what how you're saying, like using a parasitic wasp for mosquitoes or having one for like you said, ticks or anything else under the sun as well. And so they start off the paper, here are a bunch of things that fruit flies are parasitized by. And, you know, it's like, it's a neat batch of things of like what the insect hosts are and what the stages of attack there are. So there is like the adult fruit flies and what they're infected by. And here is this brand new species of this Centrenus, species novel. So that's the same species as we've, like, demarcation as we've seen before. Whenever they put SP Nov, that's the new species that they're talking about. And you can see that that fruit fly, it's been identified for that it does infect it. So there are parasitic wasps that affect these other stages in the adult form. Like we talked about here, here's some of the caterpillars that we're putting out. There is the formicae. So these are like carpenter ants. Anything that actually sprays formic acid, we've got here, that's what these are. And so that species is this Neoneurus species, is the wasp that does the infecting. I think it's a nice figure, Sam. So like if anyone is interested in general parasites and what they attack, here's actually another one too. This one's associated with Formica colonies, and there's a Sinetris here as well. So it's just, yeah, there's a couple of different ones, and it's like spaced out based on evolutionary timelines. So they do show you where the new species of the week is that they identified. And as we go further down, they have a nice little set of experiments where it's just like the physical characterization of what this looks like. Could it be more insects? Oh yeah, Sparky, listen. This blows open what we think about what can go on. The reason for it, Sparky, is if we've never described before, ever, a parasitic wasp that infects an adult fruit fly, the moment you find one, there's probably more, right? There can't just be a single individual that eats the adult fruit fly, because I guess if we think about it in a way, it is almost like the perfect host then, right? If no one else eats this life stage, then you have a total monopoly on the world. Nothing's going to let that happen. Something else is going to move into that ecological niche. So I bet you there's a ton more that work on the same pathway molecular mechanism. I already thought wasps were nightmare fuel before I knew parasitic ones existed. GG's insects. So fun fact, Shark Geek, there is, when we define what's alive, usually one of the definitions for life is like can reproduce on their own. And so it's like, well, viruses can't do that, and a virus isn't alive, so it's fine. But then you're like, wait a minute, neither can these. Like, these are, you know, these wasps are clearly alive. They're like running around, they're doing all kinds of things, right? But they can't survive on their own. They've lost the ability to reproduce without a host. Therefore, they don't fit into the definition of what is alive, like by some classic means. And so it's like both the new definition, like you have to constantly adapt because there's always these rule violators in place too. But yeah, so here is the larva. So this image here, they've taken a fruit fly adult, popped it open, and there's the wasp embryo inside. Look at that. Other ones in figs, and we accidentally, yep. But those, I believe, ecstatic, the ones that we accidentally eat, do end up being broken down by the fig itself. So I think it ends up not being like an issue, like in terms of like when you get any kind of crunchiness in the fig, it's been like reabsorbed, so it's not a worry. Hopefully, that was my that's been my takeaway of those. Yeah, but yeah, that is the wasp larva. These little dots are fat cells, so that you can actually see, like, you know, that's what they're eating. The next panel there with these arrows, these are the cells that it's like triggering and eating with the black arrows, and then this is the actual parasite right there. And then it forms, this is like the late larval stage, so L3, and then it actually surrounds itself as well with that silk for its cocoon. So that's like, they even go through really nicely the life stage of this animal. Here, Shark Geek, to add to your nightmare fuel, this is where the larva is emerging from the host. So it almost looks like it's a continuation of the body, right, with that light color, but it is not. That is the fruit fly larva, and it's digging through, boring through the exoskeleton and popping on out. Yeah, 100% like alien. Yeah, I mean, Sparky, I would say even the alien, the larval attacking ones are also like aliens, except it's a larva. This is an adult stage animal. So that was the first set. So here they did a really nice little graphical outline of what happens in their life cycle. So it's a nice little visual element, right? They show the wasp infecting the adult. The adult then, the reproductive organs are becoming more and more like these fat cells and just being released that the embryo eats. The embryo then bores out, forms a cocoon, and then hatches. And it all takes, 18 days as a larva, and then as a cocoon, 23 days. So it's got a nice little, they've got a nice life cycle timing here as well. Somewhat fascinating at the picnic that the wasp target the cold turkey meat. There are some like that too, Wombat, yeah, where they actually be carnivorous. So Sam, the way that they caught these is a standard fruit fly trap, which is not high tech at all. You take a PVC bucket and you put in a trash bag, and in the bucket you throw a couple of pieces of banana and maybe some watermelon, and then sprinkle on some yeast and let it sit for a few days. And so you wave around it, and after you hit it, and you just catch the insects in there, and it's actually adapted to go into those vials that we keep the flies in. You pop them into the vial, you put a lid on, and you take it back into the lab, and you knock them out on a carbon dioxide pad, and they all fall asleep, and you get to see what's in there. Will they infect? They do, Sparky. And I guess there's two ways to ask that. If you release them in the lab, they might infect some adults, but nothing will really too much come of it. But you can culture them in the lab. And so the big catch with this is, Sparky, that they specialize on Drosophila melanogaster, which is the common fruit fly. That is the most well-studied fruit fly in the world. All research labs that study fruit flies, maybe 2% of them that don't specialize and use Drosophila melanogaster. The reason for that is because all the genetic tools that we have that have been developed in fruit flies have been developed in Drosophila melanogaster for 100 years now. So there's just a lot of literature, there's a lot of tools, and that's why it was really interesting that they do infect those particular fruit flies. So the authors actually develop an entire protocol of how to actually go through and do the infection, which I'll show you all in a little bit. But these are the high-res pictures that we've talked about. You remember, usually with the new species of the week, there are these high-resolution cool pictures of the new species. And so we got those too. They were just looking at, so exactly. So Sam, they were looking for wild-caught flies. One of the things that you can do with wild-caught flies is just do genetic and population surveys and try to figure out what exactly is going on with each individual within a population. So Jay was asking about the success rate. Here is the success rate, Jay. So parasitoid success reached about 25% of laboratory infected and 39% of wild infected hosts. And the catch is, of course, lab infected might be genetically different, and definitely are genetically different than what's in the wild. So that's that is the actual success rate that was identified. Jay, there are male and female of these flies, which I think is pretty interesting. They survive better if they infect a female fruit fly versus a male fruit fly. They come out with bigger body size. There's increased survival. There's all this extra features. So there's something on the molecular level that's happening when they infect a female fly that doesn't happen when they infect a male fly. Females have, so Sam, that's one very good possibility, but here is the catch. Yet we observe the frequency of this wasp infection in the field is much higher in male versus female. So even though they do better in females, it seems like in the males, they're more infecting the males. And then the other thing too is they mentioned maybe it's the observation rates that's happening. There might be a bunch of these differences for it. So GK, these can't sting you. This is what they look like. It's about, let's see what this scale bar is, a millimeter. So it's 1.5 millimeters in length. Can't pierce your skin. So there is no threat. This is an adult fruit fly that has been popped open. And you can see there's some fat cells, and then there's this thing. That is the larva of the parasite swimming on around. If you look through the cuticle or the skin of the insect, you see these little circles, and those are special fat cells that are released as a function of the parasite being inside, and just kind of informs the cell hey, it's time to pop yourself and show everyone everything. And then here are the larval stage and the cocoon that ends up happening here. It does, yeah. There is like an eye like shape to it. Absolutely, GK. This is like a big deal paper, because usually these papers we look at, right? It's like they do some body measurements, they do some sequencing, and that's that. Here, yeah, they did full genome sequencing, right? So they did the whole sequencing on these animals to identify what's going on a molecular level. They did physical characterization. They also went through and identified the life cycle of how these animals survive. They also described how to catch them and how to culture them in the lab. Like, this is like, and it's also the first ever example of this animal, like a parasitic wasp infecting an adult fruit fly. It is the first example ever. Sam, so interesting point. This is on Drosophila melanogaster, which is the common fruit fly that's in your house, and it's the most commonly one studied. They did not look at a broad level of all the different species that could be infected. Yes. And so what's terrifying, GK, what is terrifying, so this is a video of the adult. So Tatum was like, how do I know if I can find them? So yeah, so Ella, here is like the way that you would end up seeing it. So this is actually a different species, Sam. So this is not Drosophila melanogaster. This is Drosophila affinis. So it's a different species than the one that they optimized the testing on. But this is like the wild-caught fly. You can actually see the testes right here in red. So you can actually sex male and female just by color of the testes. It's actually visible through. But do you see that thing wiggling? That's not the fly doing the wiggling. That's the parasite inside the host. Yeah, I see it and I wish I didn't. I know, right? No, Ella, the fly's not dead. So Ella, the background that the fly is laid on is called a CO2 pad. So it's basically a porous material that has carbon dioxide flowing through it, and it puts the flies to sleep. So you can put them underneath the microscope and do the imaging that you need to do. And so that's what we're ended up seeing here. And so I like this paper because they actually put in all these pieces there. No, at GK, these cannot sting humans. So these wasps, if they're 1.5 millimeters in length, so super small, if they land on you, GK, they'll walk around on your finger and flutter away. Like, they're not even going to attempt to sting you. Their stinger is so fragile and thin that it cannot pierce our skin. So even if they did attempt to poke at you, you'd never even feel it. You'd never feel it. Yep, anesthesia for flies. And yeah, Ella, you're 100% right. Other insects also, you can use carbon dioxide as anesthesia. The other thing that is commonly used in the lab, Ella, in addition to CO2, is ice, because you put them to sleep. But here is that wasp larva that popped out of the adult host. Look at that. That popped out of the adult fruit fly. Look at how wiggly it is. And that spins a cocoon around its body. And that's what eventually ends up being like forming the new adult wasp. Look at this pause screen, y'all. Do you see this like shank of a blade coming out from the wasp at the fruit fly? Like, that is like, whoa, come on now. Yeah, and so what's cool, I think, GK, is we actually don't know if there are yet behavioral changes on behalf of the fruit fly. Like, does the fruit fly change its behavior when it sees something like this happening? That we don't know. We might know it one day, right? If it's discussed. We know, GK, that the adult fruit fly changes its behavior as a response to parasitic wasp that infect its babies, right? So if the wasp is no threat to the adult fly, only to its babies, the fruit fly does different behaviors. Presumably, GK, one would imagine that if it could hurt you and not just your babies, you're also going to have a behavioral change. Like Sam's like, I would change my behavior even if it couldn't hurt my kiddo. Like, for me, right, I would want to not die. The wasp could have to take my money. And it's not that it doesn't happen, it just hasn't been studied in this paper, which makes sense to me. Like, the scope of this paper would have been just gigantic, and so it's easier to not have it be overstudied, right? Like, the scope of the paper, like, it's just an amazing behavior that we're able to see. And Lodok, the cocoon building happens after the larva has borne out of the body of the adult fruit fly. It just lands on the ground, and then it uses silk and spins silk around itself to basically like make itself like finished off and ready for the next stage of life. I like these videos being in this paper because it gives us a real sense of how quickly something like this happens. You know, sometimes you hear like, oh, it's infecting it, and it's like, well, what does that actually mean? How long is that gonna actually happen? Kind of a crazy amount of, like, crazy speed that we've got it at. It's not especially or closely quantified. We don't know really what these things mean. Right? Like, is there an actual behavioral difference here? We also don't know if there's other behavioral changes that also happen. This is just what was kind of put into the paper, that if the flies do perceive this threat, there's been a bunch of evidence, Andrew, in terms of the fruit flies, if you pair them with a larval predatory wasp that infects larvae, the adult fly changes its behavior to protect its babies. This is slightly different, right? Because this wasp infects the adult, not the offspring. And so there is, again, like, there is some kind of interaction going on here, but we just don't know yet to what level it means. Like, what is the end output here? Like, what is the quantifiable benefit of smacking away the wasp? Because even though, like, this fly has smacked away this wasp and told it to go away, like, the wasp keeps coming back, and presumably as soon as that fly turns around, it'll sting it, and then that's that. So, you know, I'm suspicious about how likely something like this is to work, like that, just smacking away of the animal. But it's still really cool. So if our friend Jay is still here, Jay was wondering about the success rate in terms of host success versus parasite success. And we talked about the statistics that they gave, that 25% of lab strains were successfully infected, and then 38% of wild-caught strains were infected successfully. So this is what it looks like when the parasite is not successful. So this is called melanotic encapsulation. So it is the same type of encapsulation that happens in the larval state. Different kinds of cells are acting here, but essentially what happens is that a large egg, in this case, gets surrounded by cells that are the immune cells. They release a melanin pigment, and the melanin pigment essentially kills the parasite. And so you can actually see it on the adult. Like, you can see these little dots. And those are examples of a successful immune response. So that was our first paper, y'all. I hope that y'all found it interesting. I thought it was super cool.